it's harder to breathe in the first time after that happens. Typical residual volume is a little bigger than expiratory reserve. There are also respiratory capacities, which are different combinations of volumes that tell us different things about lung function. I'm just going to cover the two major ones: total lung capacity and vital capacity. So total lung capacity is exactly what it sounds like, the entire amount of air the lungs can hold. Average is just under 6 liters, and that typically goes with your body size. The bigger your chest cavity, the bigger your lungs. And usually for men it's larger than for women. If you're taller, it's probably larger, and so on. And it tends to decrease with age as part of growing old. Vital capacity is take out residual volume and just look at the air that can move. VC for short. So if you were to take in as much air as you possibly could and then blow out as much air as you possibly could, say to fill up a balloon, that is your vital capacity. By math, it's tidal plus inspiratory reserve plus expiratory reserve volumes. Those three plus residual volume makes total lung capacity. Now, the kind of spirometer we have, air measurer, only measures exhale. So we can't measure our inspiratory reserve volume with that, so we're going to have to do it backwards. Measure tidal, expiratory reserve, and vital capacity, and then do some subtraction to find your inspiratory reserve volume. And that's part of next week's lab. Spirometry is often shown on a graph where the line going up is inhaling, line going down is exhaling. The bottom is 0, so you've got a stretch that is your residual volume that the line's never going to go below that. And that's shown with the orange arrows. Tidal volume is here in the middle in blue. So think of the tides going gently in and out. And that's your air going gently in and out. So above that would be the inspiratory reserve volume, taking you up to total lung capacity. Below is expiratory reserve volume. So if your tidal volume increased, like say you were exercising, that would in turn decrease the reserve volumes because you're dipping into them, you're using part of them. You could think of it as tidal volumes, your checking account that you pay your bills out of. Reserve volumes are the savings accounts that, if you even make enough that you can have one, you keep that there for things like car repairs or something. You occasionally have to dip into it. Any questions on spirometry? Minute ventilation, or minute volume, is the respiratory equivalent of cardiac output, where cardiac output was the amount of blood pumped in a minute. This is the amount of air moved in a minute. You get it the same way, the volume per movement times the number of movements in that minute. Here we have tidal volume instead of stroke volume, breaths per minute instead of beats per minute. For example, if we take the average tidal volume, 500 milliliters, and say 12 breaths per minute, which is on the slow end for most adults, what would we get as our minute ventilation? I think I heard it. Yeah, 6,000 milliliters, or 6 liters. Now, not all of the air coming in and out is going to get to the alveoli. Some of it is in the airways where no gas exchange occurs. Typically, there's about 150 milliliters of space in the airways. So out of a 500 milliliter tidal volume, about 350 milliliters makes it to the alveoli, where the gases can be exchanged. So, any further questions on breathing movements? Brief mention, just because I think it's interesting, of movements to your lungs using your inspiratory muscles that aren't for the purpose of getting the air in and out or ventilation. We've got speech. You take in extra air so that you can push it out through your vocal cords and make vibrations. Movements for clearing the airways, like coughing and sneezing. We also have emotional movements, laughing and crying. They both have this similar pattern of big breath and then let it out in several chunks instead of smoothly. And then some things that we don't actually understand very well, but we all do, hiccups and yawning. We know that a hiccup is a spasm of the diaphragm, but we're not entirely sure why it happens. Yawning, we used to think it was to get extra oxygen, but now that doesn't seem to be the case. And we definitely don't know why talking about yawning or seeing somebody yawn makes us do it. So I've skipped clinical application 2. Number 3 is on reduced ventilation. 2 major conditions that cause reduced ventilation are asthma, where some kind of irritation causes inflammation of the airways, and bronchoconstriction. So less air, that causes more air resistance. The air can't move in and out as quickly. Emphysema is the other one. So along with the alveolar surface area, elastic fibers are lost. And that makes the lungs have less elastic recoil. So you can inhale just fine, but the lungs don't collapse back down by themselves as easily. So with emphysema, it's harder to exhale. The next section, control of breathing, is on how we alter rate and depth. We have some voluntary control. You can hold your breath, you can hyperventilate on purpose. And then there are stimuli that cause it to change without a conscious decision. So if there's a stimulus, we can look at it as homeostasis. The control center is some respiratory areas within the brainstem, which I'll go over on the next slide. The effectors are the inspiratory muscles. How often do they contract? How hard do they contract? Thus affecting the depth of breathing. The respiratory areas are in the medulla oblongata and pons. In the medulla, there's 2 dorsal and ventral respiratory groups. So if you remember that this is the cerebellum and it's in the back, which one of these is the dorsal respiratory group? Yeah, this one, closer to the cerebellum. So the dorsal respiratory group is the main one that directly signals the inspiratory muscles to contract. So even though they're not generally voluntary as skeletal muscles, they need a nerve signal to contract. And that comes from the dorsal respiratory group. The ventral respiratory group adjusts the rate. The pons, there's a lot of research still ongoing about exactly what its role is, but it seems to process the stimuli and kind of come to a consensus and tell the ventral respiratory group everything that's going on, so it can make its decision about speed. To look at the stimuli involved in changing breathing rate and depth, we need to look at something called partial pressure. What this means is what portion of a mixed gas, like say air, is one specific gas, like oxygen, nitrogen, carbon dioxide. So if you're just talking about the portion of the gas that is, or of the mix that is that gas, you could say it as a percent, like air is about 21% oxygen. If you wanted to put a number to the pressure, you would multiply that percentage by the total pressure. So by the atmospheric pressure to get what's the partial pressure of oxygen in the air. Why partial pressure matters is that it controls the diffusion of gas across a membrane. The way most substances will follow a concentration gradient, gases follow a partial pressure gradient. Partial pressure of oxygen and carbon dioxide are important factors in breathing. Also affected by emotion, lung stretch. So for example, if you're taking really deep breaths and stretching the lungs hard, the brainstem will want to switch you over to, let's breathe faster instead and not put so much force on the lungs. Exercise, you need more oxygen, so you'll want to get more ventilation. And then blood pH is also a factor. By adjusting carbon dioxide excretion, you adjust blood pH. The receptors that send the information to the brainstem, respiratory centers, are called chemoreceptors. There are central and peripheral chemoreceptors. The central ones are in the medulla oblongata, and they monitor carbon dioxide levels. Because they're right there, they get the strongest input. So the CO2 is the most important factor in breathing rate. Peripheral chemoreceptors are found in arteries, the same locations as the baroreceptors that alter blood pressure, the aorta and the carotid arteries. They detect O2 levels and blood pH. So all three of those main factors get the input through the chemoreceptors. So if we put it together, your stimulus is O2, CO2, or pH, or maybe a combination of them. Receptors are chemoreceptors. Control center is the ventral respiratory group. Effectors are the inspiratory muscles. The response would be to change the rate and or depth of breathing. So let's take one example. Let's say your oxygen levels are low. Follow that through. What response is it going to get? Yeah, you'll take deeper and or faster breaths. Increased ventilation. Let's say your CO2 is low. Yeah, we're going to go the other way, breathe slower or more shallowly to hold on to that CO2. Okay. Now, we've got sections on gas exchange and gas transport. The lab today is kind of short, and the Monday night group finished the chapter, so I'd like us to finish the chapter, and then we'll start lab late, so everybody has plenty of time for lunch. Does that work for everybody? All right. In this section, we're going to look at a little more about the structure of the alveoli and gas diffusion. Now, in the alveoli, we have three main cell types. Type 1 cells are simple squamous epithelium. They're those little flat, kind of fried egg looking cells that make a nice thin membrane for gases to move through quickly. Type 2 cells are the ones that produce surfactants. And then there are also macrophages found within the alveoli to clean up any pathogens that make it past the mucus in the airways. The respiratory membrane is everything the gases have to pass through for external respiration to occur. So to get from air in the alveoli to blood or the other way. So that includes both the alveolar wall and the capillary wall, which is also simple squamous, you might recall, and then a thin basement membrane that holds them together. So we want these as thin as possible because the further the gas has to go, the longer it's going to take it to get there. And thus, the less of the gas will actually get through. This one's the macrophage, and this little guy here with the, I guess the microvilli, is a type 2 cell. It produces the surfactant and secretes it on all those little bumps. So, even dissolved in liquid, gases have a partial pressure, and that takes the place of a concentration gradient. A gas will diffuse from higher partial pressure to lower partial pressure. And we have arrows going both ways, blood to alveoli, and the other way, because one gas goes one way, the other goes the other way. Now, partial pressure is abbreviated in a way that I can't actually reproduce on a PowerPoint. You have a capital P and then a subscript of the gas.